Welcome. In this lecture, we're going to cover Chapter 3, the cellular levels of organization. So let's get started. Let's review cell theory. Cell theory basically states that cells are the building blocks of all plants and animals. True. And you also have to keep in mind that as the building blocks, some of them may specialize for necessary structural or functional needs. The second law states that all cells come from the division of pre-existing cells. Also, that cells are the smallest living units that carry out all physiological functions. So that means reproduction, that means respiration, metabolism, bringing in nutrients and oxygen, removing waste such as urea or ammonia, as well as carbon dioxide. Now, when we talk about cellular differentiation, this is going to produce specialized cells, which I've been talking about. And it's amazing when you think about this. A zygote, a fertilized ovum, fertilized egg cell, contains the genetic potential to become any cell in the body. And then as it divides, we go through this process of gradual specialization. This is called cellular differentiation. So at a certain point, we start forming different types of tissue, epithelial tissue, connective tissue. And as we go into chapter 4, you're going to see how there's more than just one type of connective tissue, or one type of epithelial tissue, or one type of muscle tissue. There is one particular cell that's in neural tissue, but neural tissue usually includes supportive cells. We call that neuroglia, and we'll continue with that afterwards. Usually, what happens as you go from what we would call a generalized cell to some specialization is that genes in the center, genes in the DNA, some will be turned on and some will be turned off, resulting in a differentiation that can lead it to being developed into an endocrine cell, a muscle cell, a skin cell, etc. Cells consist of basically a plasma membrane, cytoplasm, and a nucleus. Now this is for eukaryotic cells, cells that have a nucleus. But we have to consider the extracellular fluid. This is the watery medium surrounding the outside of cells. We would call this also interstitial fluid. So extracellular fluid, interstitial fluid, they're basically synonymous, okay? As I said, there are key components of cells: the cell membrane, the cytoplasm, the organelles. Now the organelles, one of them is the nucleus. We're going to explore all of them in more detail. I strongly encourage you to take the time and review some of these type of diagrams so that you can identify with a certain level of certainty nucleus, centrosome, endoplasmic reticulums of the smooth endoplasmic reticulum or the rough endoplasmic reticulum, ribosomes, mitochondria, cytoskeleton, the plasma membrane. Now, by the way, when I say so you can identify, it's not just a matter of just saying, okay, that's it. Tell me about it. What does it do? You have to understand that even at the cellular level, matters of health and disease can be influenced by either the malfunction, the loss of structure, the damage, whether it's chemical or physical or whatever, to these particular organelles. And this will result in the death of the cell and what could be eventually the death of the tissue, death of the organism itself. Let me give you one example. The mitochondria. Now, the mitochondria have their own unique loops of DNA. We'll get more into this in a little while, but I want to just take this sidebar for just a second. The DNA loops that are in the mitochondria basically code for proteins that are only inside the mitochondria. At present, there's about 30 or so diseases. Sorry about the barking in the background with my pet dogs. They're having a good fun time right now, sort of the zoomies as they call it. Okay, now here's the key point. We would have patients that would show up, they would have a lack of energy, a lack of balance or poise. They would die early. They would have multi-organ failure. No infectious agent, no virus, no bacteria, no fungi, no nothing. No environmental causes like lead poisoning, mercury poisoning, anything like that. No radioactive isotope, etc. What they did notice is that the mitochondria were dying. Self-destruct, they stop functioning, the cell stops functioning because the mitochondria produce ATP. There are about 30 or so of these mitochondrial-related disorders. Basically, the mitochondria are inherited, mostly from mom, okay, or pretty much all from mom. The mitochondria will be basically what you get from your mother's side. And what happens is that you will note that over so many years, if mom had defective mitochondria, then you will have the defective mitochondria and will have the mitochondrial disease. Now, this was, if you go back to 70s or even 60s, not really well understood because they really didn't understand all of the genetic components that make up the mitochondria. They're still exploring the genetic components. But as I said, if you have just one organelle that's damaged or malfunctioned, this could have a serious consequence to the survival and fitness of that cell and the organism that has those cells. But let's start off with the plasma membrane, otherwise known as the cell membrane. Now, in the last chapter, we talked about hydrophobic and hydrophilic. We talked about phospholipids, and what you see here is predominantly in the lipid part of the membrane. It is composed of phospholipids. There are some that are glycolipids. So instead of that round ball there, they would have basically a sugar there. You will have also these glycocalyxes, which are extracellular carbohydrates. Don't get those mixed up. When we talk about just the lipid composition of the membrane itself, you'll always notice that, excuse me, the extracellular fluid, that's the watery environment on the outside, the cytoplasm, which is the watery environment on the inside. You will have what they call a lipid bilayer. Here's one layer, here is the other layer, and you will always note that the hydrophilic part of the amphipathic molecule, this phospholipid, you'll always have the polar part facing the watery environment outside and the watery environment inside. Now, that means in the middle, this is where you have the nonpolar or the hydrophobic part. That's the barrier. And you'll notice that the yellow structures here are basically cholesterol molecules, which add to the stiffness of the molecule. Now, the cell membrane is not merely just made up of lipids. It has proteins that in some cases act as channels or transporters of substances from one side to the other. Okay? You have some that are called integral, which means that they span the entire width of the plasma membrane. You have some that are referred to as peripheral, which means that they only are either on the inside or the outside, but they don't pass all the way through the membrane. Some of these proteins are going to act as receptors. Others are going to be structural in purpose. Others are going to be adorned with saccharides. Either this would be basically what you consider a polysaccharide, basically carbohydrates that are on the outside, and they may act as a means of identification of this particular cell for cell markers, etc. Now, on the inside, you may have actually attachments, anchoring structures that play a role. They may be attached to, for example, cytoskeletal microfilaments and things like that. And so they reinforce this internal cytoskeleton structure. What you have to keep in mind is this. For anything to come through, to go from outside the cell to inside the cell, it either has to go through these protein channels, or it has to be nonpolar, not charged, small molecule. So what do these include that would easily pass through this barrier? Oxygen molecules, carbon dioxide molecules. Now, water molecules pass through, but they have to require a certain channel here. The same thing as with charged ions such as sodium and potassium. They will go through a certain channel area, okay? Otherwise, if it's a huge molecule like glucose, or even larger ones like starch, glycogen, something else like that, not going to happen. It has to be brought inside by special protein structures if it's going to be brought in at all. So the plasma membrane acts as a barrier. It's actually a semi-permeable barrier, which means that certain substances will pass through, others will not. You have the structure of the glycocalyx. You have the integral proteins, which we've mentioned. The transmembrane proteins, which pass from one side to the other. They're going to pass through the membrane, and these include pores or channels. And we also have the peripheral proteins. Peripheral proteins are otherwise called extrinsic proteins. Now, remember I talked about the phospholipids and cholesterol. Here are the cholesterol molecules. Here is the phospholipid with its hydrophobic tails. These are fatty acids. Here is the hydrophilic heads. So they will face the outside and the inside watery environments. Cholesterol tends to stiffen the plasma membrane. The term amphipathic refers to a molecule that is part polar and part nonpolar. Now, I do encourage you to review these tables or charts whenever you see them in the course. It is going to help you. There are different classes of membrane proteins, anchoring proteins, recognition proteins, enzymes, receptor proteins, carrier proteins, and channels. They're all responsible for how the cell lives, exists in the extracellular fluid, but it also helps in communication, in metabolism, and in the proper structure that you have for the cell itself. Now, we have some extensions of the membrane that are unique and specialized. One example is using microfilaments, and here we're talking about the cytoskeleton. The cytoskeleton will be made up of one of three different types of skeletal structures. Think of it as like scaffolding. It's interesting because at a certain point in time, cell biologists didn't call things a cytoskeleton because they said, well, skeleton we mean is a large, big thing, until they started getting into unique staining capabilities, and these stains would allow them to see the presence of microfilaments, intermediate filaments, and microtubules in the cell. So now you could see how the cell is uniquely stretched out. It's not a round spherical structure. Not all cells are like that. Let's go through some of the cytoskeletal elements here. Microfilaments, these are the ones that provide support for the microvilli, as you can see right here. Okay, so they kind of act as a support structure so that you can have this extension of the membrane. Now, the extension of the membrane increases the absorptive surface area by doing this. Microvilli are composed of actin, and they're usually about 6 or less than 6 nanometers in diameter. Remember, nano is billions. You would have them in some of these structures such as we call a terminal web. You can see that right here. Intermediate filaments range in size from 7 to 11 nanometers. They're the strongest and most durable. Microtubules are actually sort of, they're tubes, tube-like structures. And we see them present. They're made of a particularly unique protein called tubulin. They average about 25 nanometers. An example of them are the centrosomes, okay? Now, in the cytoskeleton, you get a variety of these remarks, location, function, and structure of the cytoskeleton elements. I encourage you to spend time and review this. The microfilaments, the intermediate filaments, the thick filaments, you can see where they're all found throughout the cytoplasm. There are some specialized areas that you will find some microtubules. For example, radiating away from the centrosome in the cytoplasm. And then we have other unique structures that are found on some cells, but not all. Centrioles are one group. That's a group of microtubule triplets. And they end up forming what's called a 9+2 arrangement. 9 triplets around and then a central one on the inside. And this is what helps form the centrosome near the nucleus. They organize the microtubules in what we call the spindle fiber structure, the spindle, so that they can move chromosomes during cell division, mitosis. Now, cilia and flagella, everybody gets them mixed up. Don't worry about that. Spend time trying to make a clear understanding of them. Cilia are shorter. They may be extensions from the cell. There are 9 groups of microtubule doublets. This is where you get, I stand corrected, this is where you get the 9+2 arrangement in cilia, not in the centrosomes, okay? So cilia have sort of a base, and they have these extensions of the plasma membrane, but at the base of them is this structure that has a 9+2 arrangement of microtubules. Now, you usually will have on a cell multiple numbers of cilia, and the cilia are mobile. They're able to basically move back and forth in a swirling, in a swifting or swirling manner, a sweeping motion. They're not unlike what you see as sort of those that are rowing, okay? And so you have those big long paddles and the rows that basically propel these small little boats. But the key point here is usually that cilia differ from flagella because cilia are basically attached to the cell and they are moving substances past the cell. For example, in your trachea, you will have cilia that are present on some cells on the inner lining of the trachea, and they move this thin layer of mucus upwards. And the mucus is used to trap particles like bacteria, dust particles, smoke particles, etc. Okay? Now, flagella, you're usually going to get 1, maybe 2. With human cells, the best known flagella you'll see is the human cell known as the sperm cell. Okay? Now, there are other microorganisms that use flagella for a variety of reasons and have more than one. But just let's focus in on human cells right now. At the base of the flagella, on the inside of the plasma membrane, you're going to have, again, 9 groups of microtubule doublets that form this cylinder around the central pair, a 9+2 arrangement. The flagella is well known as being the propulsion vehicle or the propeller. It's not really rotating, but it whips back and forth, and it's sort of the mechanism that allows sperm cells to move through fluid mediums, okay? Here you see the microtubules arrangement in a centriole. You'll notice you have the triplets here. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9. So you have a 9 set, but you don't have a central one. Here's an example of that power stroke that you see with cilia. So they will whip along, and at the base of them is this microtubule arrangement. Now, as you get higher up, you'll notice that sort of it appears to be doublets here, okay? And in these doublets, they will have a central one, and then they will have about 9 of them. There's one other thing to keep in mind. You notice that the cell membrane, the plasma membrane, rises up to the top and then comes back down, okay? Now, we move from some of the membrane structures and some of the structures that are providing cytoskeleton to something that's absolutely essential for the function of a cell, and that is the organelle that's involved with protein synthesis. This is located both in the cytoplasm, they freely float around, and there are others that are attached to the endoplasmic reticulum. This is what forms the rough endoplasmic reticulum. All ribosomes are composed of ribosomal RNA and proteins. Think of the ribosomal RNA as sort of the structure, the scaffolding, and proteins attach around this RNA. And they make a small subunit and a large subunit. When they come together, they are going to read the messenger RNA and begin to assemble this chain of amino acids that we will call the making of the peptide. We'll get more into ribosomes later on. And you got to keep in mind something too. Ribosomes, the more of them you have, the more protein synthesis occurs in those cells. Okay? Now, there are 2 types of endoplasmic reticulum I've mentioned before. Rough, which is studded with ribosomes. Basically, the ribosomes will make the protein, but they'll inject it into the inner space, the lumen of the rough endoplasmic reticulum. In that space, proteins will be modified, they may add sugars to them, and they will export them via the transport vesicles. So the inner spaces, which are referred to as tubular cisternae, here and here, exist. Smooth, though, if you notice, there's no ribosomes here at all, okay? Here is a re-rendition here. Here's our cisternae here for rough endoplasmic reticulum. You have the messenger RNA strand, you have the ribosomes, and they're making this growing polypeptide, and eventually it's fully secreted and released into this luminous cisternae space where it may be modified, it may be fixed, arranged, and eventually it's purified and sent out by transport vesicles. Okay? But then we have the smooth endoplasmic reticulum, which does a lot of unique and very important cellular functions. It is involved with the synthesis of phospholipids and cholesterol needed for the maintenance and growth of plasma membranes, endoplasmic reticulum, nuclear envelope, and Golgi apparatus. It is involved with the synthesis of steroidal hormones such as testosterone, estrogen. Okay? Especially you're going to see this in reproductive organs. It is involved with the synthesis and storage of glycerides, especially triglycerides. You'll see this particularly in liver cells and fat cells. It is also involved with the synthesis and storage of glycogen in skeletal muscle cells and liver cells. Now, the Golgi apparatus is a little bit different. What you need to understand is this. Just like you have a shipping department, you have a receiving dock and you have an exporting dock. What do I mean by that is, remember those transport vesicles that left the rough endoplasmic reticulum? Well, here they are, and they're picked up by the cis face of the Golgi apparatus. It is also known as Golgi bodies. And what happens is this material gets bopped around back and forth, purified, concentrated for one particular substance here, and then it bubbles off in another membrane-bound vesicle. It may be heading off to provide enzymes for the cytosol as a lysosome. It may be bubbling off and eventually will go through a process of secretion here where the vesicle fuses with the cell membrane and releases its contents to the extracellular fluid. Okay? Also, you will get from time to time, you will bubble off something, but basically what it is to replace membrane that has been damaged or lost. And that membrane will also contain, obviously, some other proteins which have been made previously, but then packaged and implanted into the basically the cell membrane wall here. Okay? Now, so cis is the receiving side, trans is the exporting side. And what about these lysosomes? This is really strange. Could you have a substance that contains some of the most powerful enzymes that dissolve all sorts of lipids or proteins or whatever? Well, think about this. In alchemy, there was a sort of a concept. One of them was to make an acid that would dissolve anything. But that gets to be contradictory, doesn't it? An acid that would dissolve everything, how would you hold it? What kind of container could you put it in? Well, digestive enzymes don't dissolve everything, but they do have to be put in a specific site, encased in a membrane. Now, there's a reason for that. In part, if you just basically secreted or leaked out these enzymes everywhere, they would begin to start destroying other cellular components, cytoskeleton, other organelles, things like that. The basic functions of lysosomes are to do the following: destruction of foreign material, autolysis, and removal of damaged organelles. Let me get back to autolysis last, but let me start off with foreign material. Is some substance, solid, a fluid, basically a food particle, etc., gets brought inside by endocytosis, you now have this very interesting little membrane-bound structure. But you got to break down what's in here. So that's where the lysosome fuses with this membrane-bound structure and breaks down the components, proteins to amino acids, triglycerides to fatty acids and glycerol, polysaccharides to just basically monosaccharides, etc. These will leak out of the vesicle. Sometimes, though, some cells will globe something, let's say like a bacterium. And let's say that this is a white blood cell. After you've destroyed the bacterium that's in here, you basically will expel it later on. Now, here's the tricky part, because if this membrane-bound structure, which first starts off with inactive enzymes and eventually gets activated, if you set a very fine needle into and puncture this vesicle and allow the lysosomal enzymes to leak out, it will destroy the entire cell. Remember, enzymes can be reused time and time and time again. Lysosomal enzymes can be used to destroy damaged organelles. But one of the other things that happens is that when lysosomes are triggered under certain cellular stimuli, they will begin to leak out their contents and the cell will die. This is part of a process called apoptosis, programmed cell death. Okay? Now, membrane flow. This is where we refer to continuous movement and exchange of cell vesicles within the cell. With the exception of the mitochondria, all membranous organelles in the cell are either interconnected or in communication through the movement of cellular vesicles. This dynamic process allows cells to exchange lipids, enzymes, receptors in response to specific environmental stimuli. So, if some situations occur and we need more receptors, etc., we will crank up the protein synthesis of receptors. One thing that you're always going to understand is that cells do not waste making proteins or any other substances that they do not need at the time for the cell to function. At that, let's move into another interesting organelle, and it's called the mitochondria, which I've mentioned before. The mitochondria has been referred to as the powerhouse of the cell. It is a double membrane structure, yet interestingly enough contains its own DNA, ribosomes, and it seems to code for just proteins inside here. It's believed that an ancestral cell was enveloped and brought inside, and the ancestor to the mitochondria was the cell that was brought inside. And so they set up a symbiotic relationship. You give me energy, and I will give you shelter. You give me shelter and nutrients, I will give you more ATP per molecule of glucose. The mitochondria, interestingly enough, provides for two major processes. They're involved in the synthesis of a lot of ATP molecules. The citric acid cycle, also known as tricarboxylic acid cycle, and the electron transport chain. And all of these are located inside of the mitochondria. As a result, you get a lot more ATP out. You will also get, as part of cellular aerobic respiration, the production of H2O and CO2. Okay? So you need to be aware of the double membrane, the outer membrane, the inner membrane, these invaginations that we call cristae. And on the membrane itself are lots of enzymes involved with the synthesis of ATP molecules. And on the inner part here is called the matrix. Okay? All right. As I said, we have ATP synthesis, but you want to keep in mind that this requires oxygen. That's why we call this aerobic metabolism. Cells that don't get their oxygen cannot use the processes that occur in the mitochondria, because part of the processes require that oxygen be present amply to help drive the citric acid cycle, and eventually that helps to drive the electron transport chain. In the cytosol, you get just a little bit of ATP molecules made per molecule of glucose. Okay? Now, when we look at the nucleus, that's really the control center of the cell. It controls homeostasis for the cell. It controls protein synthesis. Basically, all the DNA that's going to be coding for most cellular activity is going to be inside that nucleus, and it doesn't go anywhere else. If you need to make a copy of that DNA instructions, you make it in the form of messenger RNA, and that's shipped outside the cell. But for all practical purposes, DNA is not supposed to leave this membrane-bound structure we call the nucleus. Okay? If we have changes in the extracellular fluid, we're going to note this through the binding to membrane receptors or diffusion through the membrane channels. These may cause changes in genetic expression because there will be nuclear receptors, and these are sort of like little proteins that grab onto whatever is being exported, brought into the cell, and they will alter the genetic activity. They'll either turn it up or turn it down. Short-term adjustments, basically it's a matter of enzyme activation or inactivation. Long-term adjustments to changes, and this is how we get homeostasis, guys, for the cell, are usually done by either biochemical changes of the cell or actually genetic changes that lead to these biochemical changes, or may actually lead to changes in the physical structure of the cell. Now, let's talk a little bit more about the structure and function of the nucleus. It is a double membrane for its envelope. This is sort of the membrane that contains everything on the inside. We have what we call the nucleoplasm, similar to cytoplasm. It's fluid inside here. And we have the nucleoli. If you stain the cells, usually if you can stain it so it stains the nucleus, the nucleoli will take the stain even further. When we talk about staining of a nucleoli, what is a nucleoli? It contains RNA, enzymes, and histone proteins. It is usually the site where ribosome components are assembled. Okay? Now, nucleoli are very prominent in cells with lots and lots of protein synthesis. So you're going to find nucleoli present very prominently in liver cells, muscle cells, and nerve cells. And by the way, if this is a double membrane, how do you get substances out? Well, this is where we get into what they call nuclear pores. These are small gaps created that are very selective. Substances can come in and out, but under certain criteria only. If we have DNA inside, it's called chromatin. Now, chromatin can be really tightly wound up, condensed, or unwound. Unwound DNA allows for expression or basically reading off the DNA instructions so that you can make a copy of that information in the form of messenger RNA and export it out of the cell. But how do I compact that? Well, think about this. If I have a big long piece of rope and I want to walk from room to room with it, do I just kind of grab one end and just trail it along? Nah, you can get lost and broken. It get tangled up. It's better if I coil it all up. If I coil up 6 feet of rope, I can make it into a nice compact little package and lift it and move it from one place to the other. So how do we coil up DNA? We coil it up by a process of wrapping it around these little clusters of proteins called histones. Now think of it just like a loop of string around a spool. If you've ever watched when they're installing new wiring, the wire is wound around a very large round circular structure, a round structure that we call a spool. The more that you coil the DNA, you will not get DNA expression there, but if you coil it and coil it and coil it, and then coil it and coil it further, you can basically take a very long strand of DNA and wrap it around each one of these histone spools to the point where you get it into a nice compact structure. This nice compact structure we call a chromosome. This is how you're allowed to enhance the storage of long DNA strands. As you can see, here's our dividing cell. Here's our visible chromosome. Now look, as you can see, it looks like it's been kind of tightly packed up, right? And if you look at this even closer, you see this, what they call supercoiled region. And you see off from the supercoiled region a lesser coiled part that basically eventually gets to the point where you can see where that loop of DNA is wrapped around all of these clusters of histones. Now, histones, a functional histone is really made up of 8 subunits. So you need to be aware of that. Prior to cell division, DNA coiling has to occur, and it becomes tighter and very complex. So you basically have these visible chromosomes. They contain a large amount of DNA, but it's all tightly wound up in a very small packet we call a chromosome. How do we take the chromosomes that basically match each other? In other words, this is chromosome 1, this is chromosome 1. They're basically copies of each other. But how do we basically transport them? Well, there's a structure here called centromere that basically adheres both of them together. And so what we have is chromosome 1 and its copy, because remember, before we go through cell division, we've got to make a copy of all the DNA, okay? Keep in mind that a normal human cell has 23 pairs of DNA. Prior to cell division, just before the cell's going to start going through this mitosis, you actually have 46 pairs, 2 sets of 23. And that's normal, because when you say a normal cell has 23 pairs, Well, just before we're going to divide that cell, we've got to make a copy of all those 23 pairs. That leads us to 46. When we have 46, we then separate the cell, separate all of the pairs so that one cell, one daughter cell gets 23, the other daughter cell gets the other 23 pairs. Now, there is a concept called the central dogma of biology, and it basically goes like this. DNA leads to RNA, which leads to proteins. The direction of basically the information, if you want to say it, information equals protein sequence. Okay, the sequence of amino acids that make up the protein. You're basically saying that DNA is really has the code for that information. But like anything else, we don't send the original map out into a place where it could get damaged or anything. Rather, we make a copy of it, and we ship the copy out. And that copy is the RNA. Then RNA is translated basically from a sequence of nucleic acids to a sequence of amino acids. The amino acid sequence, or the amino acids that are assembled together, make up the proteins. A gene is a functional unit of heredity. The chemical language of protein sequence is the genetic code. Each triplet of the DNA code, so each triplet meaning 3 of those nucleotide bases, codes for a specific amino acid in that protein sequence. Now, we're heading down the road to protein sequence. Why? Because it's important for us to understand how we get from just saying, okay, these are what the organelles are, to how do we have the cell maintain itself based on the fact that we know the nucleic acid is inside the nucleus. How does that information go from step 1, the nucleus, to step 2? You make a copy of that information in messenger RNA. Step 3 is it leaves the nucleus. Step 4, we now read that information via the ribosome and translate it into a sequence of amino acids. Okay. Well, this is where it gets a little interesting, because if you notice here, here's the triplets that we were talking about. So this is a triplet of A's, adenine, adenine, adenine. The DNA triplet coding strand is really thymine, 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 because A's go with T's. Now, when you get it into a messenger RNA, it comes out as U's. Okay, so we start with A's, we go to U's. There is a structure called the transfer RNA. The transfer RNA brings in a specific amino acid, but it has a particular triplet area that has to match, very briefly, with the RNA codon. In this case, it would be AAA, which can basically base pair with the UUU, and the tRNA that does this is bringing in the amino acid phenylalanine. The same thing goes with, here is the DNA triplet template strand for AAT. It's going to be now with RNA. Remember, wherever there's a T, you've got to have instead a U. So if it's AA here, you're going to have UU. And if this is T, of course, you're going to go back to A. The transfer RNA anticodon is going to be coded AAU. They will briefly have a base pairing, and they will deliver leucine. Now, there are some videos for this, and I encourage you to take time to view them. This will help illuminate you on these issues. Now, let's look at the bigger picture, going from DNA to proteins. DNA is a double-stranded structure. But first, for us to get into the information, which is really in the middle there of those nitrogenous bases, we're going to have to separate the 2 strands. And we now separate the 2 strands to get at the nucleotide sequence. And this is all due to gene activation. In other words, you're not going to get at this information randomly. You have to have some activation event that occurs, And then you will begin making the process of unwinding or separating the two strands of DNA so that you can start transcribing or copying the DNA code to RNA. Now, to help you, because people get translation, transcription mixed up all the time, you're not alone. If you hear me even slip once in a while, bear with me. But the way that I tried to think this out is this. When you transcribe something, you're what? Making a copy of something. Okay? So I'm taking the DNA code and I'm transcribing, making it a copy, but it's into another form of a nucleic acid. It's in the form of RNA. RNA leaves the nucleus. The RNA code, which is a nucleic acid code, is going to get translated, converted from the language of nucleic acids to the language of amino acids. You're going to get the amino acid sequence. Now, you have to understand that there are enzymes and ribosomes that assist in these processes. Okay? So, from DNA to proteins, a summary is basically this. The DNA triplets determine the sequence of messenger RNA codons. The messenger RNA codons determine the sequence of the transfer RNAs, which come in to bring to the ribosome the specific amino acids. The sequence of transfer RNAs determines the sequence of amino acids in the polypeptide or the protein. There are 3 versions of RNA, so if it gets a little confusing, you have to look at what the little letter is before the RNA letters itself. rRNA, ribosomal RNA, provides the framework for the ribosome organelles. That's the scaffolding to which all the proteins that help make up the big globular structure of the ribosome, they all attach to. Messenger RNA, mRNA, contains the DNA code for the protein sequence. Transfer RNA, tRNA. This transfers the amino acid code for unique RNA codon to the ribosomes for synthesis if the protein strand allows for it, okay? To help you, this is the anticodon part of the tRNA. Notice that it's having an amino acid attached here. This is the messenger RNA strand. This is its particular codon here, 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 and here. Notice that the codon, okay, you're going to have a base pairing sequence where if it's U here, the anticodon's got to have an A. If it's A here, the anticodon has got to have a U. If it's G in the codon, then the anticodon's got to have a C. And the same thing, if it's a C in the codon, it's got to be a G. All right? Now, we talked a few minutes ago about transcription from DNA to RNA. As I said, you have to have certain enzymes that play a role in doing certain functions. First, the DNA has to be unwound to access the nucleic acid sequence. Remember how it's the double helix? And so if you even straightened it out, you have 2 strands here. And remember, there are hydrogen bonds in between each of these nucleotides, okay? The enzyme RNA polymerase reads the DNA here and then copies that sequence into messenger RNA, okay? Now, the messenger RNA is being scooted out here, okay? And what will happen is a messenger RNA strand where you have, of course, codon 1, codon 2, et cetera, et cetera, et cetera. When the messenger RNA as it's going through is complete, it hits a particular codon called the stop codon, it'll pop off. The messenger RNA strand will pop up always. And as I mentioned to you a couple of times before, in RNA, wherever there would have been a thymine, it's replaced with a unique nitrogenous base called uracil. uracil will base pair with adenine. Now, here's an interesting point also. When they first were taking out some of the RNAs from a cell, they noticed that the RNAs were not as long as the actual genomic DNA. In other words, the DNA that was still inside of the nucleus. So they start scratching their noggins and going, well, how different is this? And through some molecular biology techniques, they were able to deduce that this mature messenger RNA, as they would call it, outside of the nucleus is shorter. What? Yeah, there's parts of it that are missing. This is where we get into a issue of removing introns and exons. This is sort of a process that takes the messenger RNA that's still in the nucleus. They call it the pre-mRNA, and it's edited. And this is before it leaves the nucleus. And in the editing, it removes the introns, which are in this lighter color here, B, D, F, and H. But at the same time, this particular cluster of little proteins that acts will fuse together the parts that are retained, which are called exons. These are the coding sequences that are spliced. They'll be put together, A, C, E, G, I. Once that this is all done, and you have the mature messenger RNA, this strand of messenger RNA will exit the nucleus from the nuclear pores. So we start off with DNA, we make a messenger RNA strand, we adjust for it, and then we expel it. Now, once it gets out, whether this is in the cytoplasm or attached to the rough endoplasmic reticulum, you have three primary steps by which the messenger RNA comes together with the ribosome. You have initiation, elongation, termination. Initiation is the process which forms the initiation complex. You have the smaller ribosomal subunit. You have the start codon here. The messenger RNA strand is pretty much throughout the length of it, so it starts. But before we really even get going here, we've got to also add our transfer RNA right up here with its first amino acid, and then capped on top is the larger subunit of the ribosome. Okay? So this forms the initiation complex, the anticodon from the tRNA and the codon from the mRNA bind together right there. The next step is elongation. This is where literally the ribosome moves along the messenger RNA strand. So it's going to go ka-chunk, 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 and the messenger RNA is going to pass through it. Every place that you've lost your tRNA, that gets kicked out. There are other enzymes that are involved to add on a replacement amino acid for subsequent use. And eventually you come to the termination part, which is where basically you come upon a stop codon. And when basically the ribosome gets the stop codon, everything falls apart. The protein is released. You have supposedly a releasing factor that plays a role also this. And what happens is the larger ribosomal subunit, the smaller one, they separate out. You've got your completed polypeptide, and the messenger RNA will then float around. It may be used again, or it may be broken down by enzymes. Okay, so you can see this. Here you have step one again. You started off. This is initiation. You have the capping with the large ribosomal subunit, and you move along. Then you have basically elongation. As you are attaching more amino acids, you're finding that you have an A site, an E site, a P site, and of course, as you lose your amino acid here, from here to here, this will be kicked out, and this will move in this direction. The ribosome will move in a direction downstream, continuing to expose and read off the codons. and you will have the anticodons from the transfer RNA coming in, attaching and also donating another amino acid until you come to termination. And that's when everything falls apart again. Now, I've moved along here for gene expression. What we do need to talk about are some cell membrane properties. As I mentioned to you earlier, the cell membrane is a semi-permeable or selectively permeable membrane. Okay? There are different types of permeability of membranes. Some of them are impermeable, nothing gets through. Freely permeable, everything gets through, or selectively permeable. Certain substances will get through. Plasma membrane is selectively permeable. If you notice, you have these different substances that vary in charge, size, and basically whether they're hydrophobic or hydrophilic. Carbohydrates are relatively large, so is the protein. Lipids will pass through any lipid membrane. Ions are charged, so they won't easily pass through or won't pass through at all the membrane. Water will pass through, but only because it has certain selective channels. They're called aquaporins. We'll get into that in a minute. Now, when we talk about molecular transport across membranes, we have to consider that a plasma membrane is really a barrier. It's going to selectively restrict diffusion. Lipid soluble, nonpolar, non-ionized, small molecules can easily pass through. Oxygen, carbon dioxide, things like that, they're going to easily pass through. But other molecules are either too large, ionized, polar, which means that they're lipid insoluble, and therefore they cannot easily or cannot pass across at all via diffusion. That's where we're going to get into membrane transport now. Membrane transport can be divided into passive and active. Passive is going from high concentrations to low concentration. Now, in the lab, we talked about this. Basically, it's almost as if you take some perfume, you put it into one certain site in the room, the rest of the room doesn't have any perfume, and after a while, you have this simple diffusion, the spreading, where you have a concentration gradient where the perfume is, that's a high concentration, and the low concentration is the corner of the room where there is no perfume. But after a while, the molecules spread out by diffusion and eventually get to those places where there is no previously perfume molecules, okay? The key with passive transport is that there's no energy required, not a bit. Now, there are different types of passive transport. Simple diffusion, such as I gave you that example. Osmosis, which is the transport of water molecules, but usually what you have are unique molecules called aquaporins that are going to be integral proteins. They're going to span the entire width of the cell membrane that allow water molecules to go in or out. And then we have facilitated transport. This is also referred to as carrier-mediated transport. These are the type of proteins that are integral. They can be either channels or carriers. Depending on what type of molecule, they will allow them, though, only to go in the direction of from high concentration to areas of low concentration. Active transport. This is different. This is going from low concentration to an area of high concentration. Okay? Now, if you have a difficulty with that, think of it this way. Passive is going from high concentration to low. It's just like you're on the top of a hill and you push a boulder down the hill. It's going to go boink, 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 boink. Okay, fine, easy. Active transport, you have to push the boulder up the hill. That's a lot tougher because that's what's going to require energy and sweat, and you'll be sitting there about halfway up going, what am I doing? Oh my gosh. This is where we use certain proteins that are called molecular pumps. Just like if you had to pump water out of your basement. What do you need? Well, you need the pump. Okay, what else do you need? Electricity. is that pump has to move the water from an area of low elevation to a higher elevation, get it out of the house. So we're going to go through a little bit of this. In extracellular fluid, you have materials that will cross the plasma membrane through active or passive processes. Okay? At the plasma membrane, passive processes do not require energy. In most cases, it's going to be in the form of ATP. You could have simple diffusion where it goes right through the plasma membrane, or you can have carrier-mediated. Now, when I say carrier-mediated transport, these are proteins that are unique in that they will allow either they will grab onto or they'll become tunnels for unique molecules that may be either ions or certain molecules of smaller sizes like glucose, etc. Okay? Active processes require ATP, and they will move substances from areas of low concentration to areas of high concentration. That's why they require energy, and usually that's where you're going to have either carrier-mediated transport or vesicular transport for large items. We'll get more into this in a minute. Let's go over an example of diffusion here. Diffusion is driven by thermal energy of molecules. This is, of course, your little dye cube dropped into a pure water. In the beginning, you see an area of high concentration here of dye molecules, areas in the water of low concentration. After a period of time, some of the dye molecules begin spreading out, going from areas of high concentration, spreading outwards to areas of low concentration, until you have an equilibrium where all spaces have dye molecules. So there's basically no longer a concentration gradient. Once you have that, you have equilibrium because the distribution of the molecules throughout the container is even. The factors that affect diffusion, though, are distance, molecular or ion size, temperature, concentration gradient, and electrical forces. Now, let me go through these basically relatively easy. When you think about this distance, the shorter the distance, the more quickly the concentration gradients are eliminated. If you're looking at transporting through a space of maybe about 1 millimeter, that's really quick. It'll be bing, done. If it's half, let's say it's half, no, let me use metric. If it's 1 meter, it takes longer to go from one end of that meter stick to the other. But once you have eliminated the concentration gradient and you have equal distribution in all parts of that transport area, that's it. There's no movement net-wise. In other words, what I mean by that is nothing else is going to go really far or go in any particular direction. Now, molecular or ion size, smaller molecules or ions move faster as they travel through the medium, okay, for simple diffusion. Temperature, the higher the temperature, the faster the diffusion rate, okay. Concentration gradient, the steeper, that is, the greater the difference for the gradient, the faster diffusion proceeds. So if you've got one little area that's got an extremely high concentration and the rest of, let's say, the room has no concentration, that's a very steep, great difference. And so the molecules will travel faster throughout the entire space of that room. Finally, electrical forces. This is where we find that opposite charges attract. Same charges repel. The attraction or repulsion can affect ion diffusion rates. If you're basically moving from one side of the membrane to the other, and all of the ions that are present, let's say, are all positively charged, you're really not going to get a lot of transport because it's going to be hampered by same charge repulsion. But if you had across a membrane lots of positive charges and on the other side lots of negative charges, you're going to have movement of the diffusions across very, very quickly for negative to positive or positive to negative. Okay. And here you see the simple diffusions as I've mentioned before. Lipids, the lipid-soluble molecules, water and soluble gases such as oxygen, carbon dioxide can easily diffuse across a lipid bilayer of a plasma membrane. When you have larger molecules, you have to have these channels or transporter proteins that will allow you to move from higher concentration to lower concentration. Water and small water-soluble molecules and ions diffuse through membrane channel proteins. But you see the membrane channels, these tunnels here, will vary in shape, size, and specificity. There are some that are unique only for sodium ions. There's others that are unique only for potassium ions. Sodium and potassium, even though they're ionized, are atoms of very different sizes. Sodium is usually smaller, potassium is usually much larger, okay? Now we get into the U-tube, okay? And in the middle of the U-tube is a selectively permeable membrane. So we can play around with this for a bit because we have to deal with the issue of osmosis. Osmosis is the diffusion of water molecules across a selectively permeable membrane. The concentration of water, the solvent, remember we talked about a solution is made up of both solvent and solute. When we talk about the concentration of water in this case, this is affected by the concentration of the solute. If you have absolutely pure water, all you've got is solvent, no solute. If you have mixtures that are, let's say, 10% sodium chloride, so 10% saline solution, you've got 90% water, which is the solvent, but you have to deal now with the other 10%, which is really the sodium chloride. And as we know, we've talked about in the previous class, sodium chloride will dissociate not into one particle, but to two, the sodium ion and the chloride ion. When we talk about the term osmotic flow, this is the movement of water driven by osmosis. When we talk about osmotic pressure, this is the force of pure water that moves into a solution as a result of its solute concentration. So if you take a look here, what do you have? You have two different concentrations. Look at the number of large particles here. These are the solute molecules, and the little dots are the water molecules. The water molecules can pass through the selective membrane, but the solutes cannot. So what's going to happen is this. Here, I have a hypotonic solution. Here, I have a hypertonic solution. So what's going to happen is the volume gets decreased, and what it is that the water will migrate out over to here because water will follow wherever there is less water. In essence, you have high concentration of water here, lower concentration here, until you receive this equilibrium where the concentration of water and solutes is equal here as it is here. Now, if you look at this force that was going through, you can apply an equal and opposite force here to get what we call the osmotic pressure. In other words, what was the force that was coming through here? Well, if you use a weight and you do it right, you can equalize it and force the volumes to still remain equal. But you have to have this force equal the force of the water that was coming in through here. And if you do it right, it works out really interesting. Now we get into the concept of osmolarity or osmotic concentration. This is a concept where the total solute concentration in an aqueous solution exists. Osmolarity, we're putting emphasis on the solute concentration. We recognize that our solvent is water, aqueous. And we have to get familiarized with certain terms. This is where you get into osmolarity, the concentration of solutes, tonicity. This is a description of how the solute affects the cell. This is usually by comparing solutions inside and outside of the cell. We started getting into this a little bit in the lab. When we talk about hypertonic, that particular concentration has a greater concentration of solutes. When we talk about isotonic, we have, when we compare inside and outside solutions, they both have the same concentration of solutes. When we talk about hypotonic, we're talking about a concentration of solutes compared to some reference, let's say inside or outside of a membrane, whatever, it's less. Now, this is the problem. Why is this so important? Let me tell you. Here's a couple of thoughts. Why is it that I can't inject an IV line pure water into somebody's vein? We'll get to that answer in a second. Why is it that if I pour salt onto the surface of the soil where a weed is, it will kill the weed? Finally, we get into one other concept. You may have heard of the rhyme of the ancient mariner, and there's a part in it where he sits there and says, "Water, water everywhere, and not a drop to drink. Water, water everywhere, and the planks did shrink." What you really have to understand is, first, we do not inject pure water into someone. You put it as an isotonic saline. If you injected pure water into a vein, the red blood cells surrounded by this pure water would have literally, basically you would have a hypotonic solution, and then the fluid on the surrounding these red blood cells is hypotonic, and inside those cells would be considered hypertonic. So the water would attempt to go into the cells to dilute the concentration of solutes inside of the cell. The problem is that cells have a certain volume limit, and if you exceed it, the cells burst. That's called hemolysis. Now, the next one was pouring salt onto the soil to kill a weed. If you have a high concentration of solutes surrounding something like a plant, what's going to happen is the roots, instead of taking up water, will be releasing water back into the soil because of the higher concentration of solutes and the low concentration of water present. As a result, the water coming out of the plant will cause the plant to shrivel up and die. Here's another one: water, water everywhere, and not a drop to drink. Well, everybody knows that if you're stuck on the ocean, you don't drink ocean water. It contains a tremendous amount of salts, not just sodium chloride, potassium chlorides, and magnesium, and all these other electrolytes, salts. And what would that do? That would strain your body, and your body would work on expelling the salts by making lots of urine. And by doing that, you then become dehydrated. Okay? What would happen if you took a very high concentration solution and injected it into the blood is that now you have a hypertonic solution surrounding the blood cells. Inside of the blood cell, although they have a lot of solutes, they would have more water inside than outside. The water would go from inside the red blood cell outward, and the cell would lose volume and begin to shrink and look shriveled and wrinkled up. That's called crenation. And if you look at this, you can see this very clearly. Isotonic, same concentration inside and outside of solutes and water molecules. Although you have water going in and out, it's all balanced out. If I take a red blood cell, put it in with a hypotonic solution like pure water, the water rushes in, the red blood cell loses its biconcave shape, fills up like a balloon, and then bursts. That's not good. If you had a very concentrated solution on the outside, it could be so laden with solutes that the concentration here is hypertonic compared to the inside of the cell. So water molecules would migrate out, the volume would decline, and the cell would shrink up and shrivel up. And that's what you have with this called crenation. It's called a scalloped look. And I give you also these two concepts here. One was the rhyme of the ancient mariner. Oh, by the way, they said, and the boards did shrink, the planks on the floor. What happens is that if you have salt water there, the boards have still some water inside of them. The water would migrate out and it would warp the boards that make the planks or the flooring of the ship. Okay, let's move on. Now, the next area we're going to have is what's called carrier-mediated. So, in other words, you have something that acts as a protein that assists or facilitates the transport across the membrane. Okay? So, molecules that are too large or insoluble in lipids have to be assisted, that is, facilitated across the cell membrane. Carrier proteins, these are transmembrane, otherwise known as integral. They span the entire length, or width, I should say, of the cell membrane. They will move one substance across the membrane from one side to the other, whether it's inside to out or outside to in. Now, some proteins, interestingly enough, carry two items across the membrane. Now, do they take two items and go from outside to in or inside to out? Well, if they do it together, then it's known as co-transport. But there are some transport proteins that do one inside going to out and at the same time take one from the outside and go in. Okay? And this is called antiport. Another name for movement in one direction, they're called symport. The movement in the opposite direction also goes by the term countertransport, and the protein is called an exchange pump. An exchange pump. The best known, sodium-potassium exchange pump. Now look, this one you absolutely need to get down. Know it cold, hard, and fast. Why? Because it's the reason that we are all alive. It is one of the basic ion transporters that maintain the balance of ions both in the extracellular fluid and in the intracellular fluid. This transports ions against the concentration gradient. So let me help you here. Here's cytoplasm, here's extracellular fluid. These darker spheres are sodium ions. These orange-colored spheres are potassium ions. Look at, there's more potassium that is always on the inside of the cell than on the outside. Same thing, there is more sodium on the outside than there is on the inside. But sometimes you have some ions go inside or outside, such as in the conduction of a action potential, which we'll get into later on in the course. And so you have to reestablish these concentrations. So you're going to have to pump it against a gradient. Pumping against a gradient means you're an ion pump that requires energy, and that energy is in the form of ATP. So you get some potassium that's on the outside, you pump it back in. It's low concentration here, it's high concentration here. Same thing with the sodium. The sodiums get pumped from a low concentration here to a higher concentration here. Again, you're going to need energy in the form of ATP. This is going to create the hydrolysis of ATP to ADP and what's called PI. Now, PI stands for inorganic phosphate. It's just a phosphate molecule, a P with oxygens around it, and yet there are certain enzymes that would allow you to reverse this process to take ADP, add in the PI to form adenosine triphosphate, ATP. This has also been referred to as the sodium-potassium ATPase. So I want you to be aware of both names. Sodium-potassium exchange pump, sodium-potassium ATPase. Now, a secondary active transport uses sodium concentration gradient and has glucose tag along. There's no need for ATP. Here you can see the glucose coming in. At the same time, the sodium comes in. Because sodium is greater out here than it is in here, what will happen is that as these two molecules, well, the glucose molecule and the sodium ion bind here, this protein will flex. And as it flexes, it will dump out sodium ions to the inside, as well as the glucose. No problem if we have sodium here because we can pump it back out using the sodium-potassium pump. Now, molecules are transported inside and outside the cell in membranous sacs. This is where we get into larger molecules and transport of them. For example, endocytosis. In all cases, endocytosis and exocytosis, they're all going to require ATP for energy. Endo means an inward direction. Exo means going out to help you. So, endocytosis, you have an inward direction. The vesicles formed are called endosomes. You have a receptor-mediated endocytosis, as well as several other lesser, well, better known, but of less complexity, and that is pinocytosis and phagocytosis. But let me deal with receptor-mediated endocytosis first. This is where materials bind to cell receptors on the membrane. Now, on the inside of that membrane, it's coated with a clathrin, which is a type of protein. And what happens is it starts to form a crater, invaginate inward. As all of these receptors that have their materials bound to them, they all kind of get cratered in and eventually encapsulated in and brought inside of the cell. That's going to be a very fancy form of endocytosis, requires ATP. The two examples of how that occurs is one, insulin, and the receptor would be insulin receptor. The second example would be cholesterol, and there would be a cholesterol receptor that binds, and that's how the cell brings in cholesterol molecules. Pinocytosis is very simple. What you basically have is the extensions of the cell around a liquid, and this has been referred to as cell drinking. That's pinocytosis, cell drinking. Phagocytosis is known as cell eating. Phago, to eat. And when instead of engulfing a liquid blob or whatever, it's engulfing some solid large material. It could be a bacteria cell. It could be a large protein. It could be any of a number of other larger molecular structures. When you form this membrane around this structure and bring it inside the cell, you have what is called a phagosome. Now, the interesting thing is to stretch out the cell membrane to engulf and bring it in requires what's called pseudopodia. Now, pseudopodia is sort of like false feet. They're extensions of the cell membrane. They are used to engulf and trap and encapsulate that particle, and that encapsulation, and that's going to be a vesicle that we call a phagosome. Exocytosis is sort of the outward transport, which I'll show you in a minute. If you start here, this is the receptor-mediated transport, and you can see where on the extracellular fluid you have these particular ligands. Now, ligands is just a generalized term for a particular molecule that binds onto a receptor. Here we have these dense setup of receptors here, and when you start having the ligands bind to them, what's going to happen underneath is this clathrin protein will begin to form the pocket. They're all these receptors with bound ligands are going to basically cluster together, form sort of a cavity. Eventually, it forms almost a endosome because the pockets are going to pinch off from the cell membrane. And there you have a clathrin-coated vesicle. The clathrin eventually detaches and is recycled. This particular vesicle may fuse with a lysosome, which is filled with digestive enzymes, and that the digestive enzymes will free up the ligands and break them down. The rest of the the rest of the receptors will eventually basically be transported back as a secondary, as an endosomal structure, and you will get them to eventually fuse with the cell membrane and reuse those particular receptors, okay? Now, does this occur? Do we actually see this? Yep, you can. Here's an example of pinocytosis here. Here's the bloodstream up here. This is a transmission electron micrograph, so it's at 20,000 X. Here is the cytoplasm. Here is the plasma membrane, outside, inside. the surrounding tissue. This is basically of a, if I'm not mistaken, an endothelial cell. And so what's happening is some of the fluid is being taken inward by endocytosis. See these bags here, and they will be transported across and then released to the other side, to the surrounding tissue. Now, I talked about phagocytosis, and people will sit there and say, so? Well, when you deal with immunology and you deal with the immune system, we see the specialized cells that perform phagocytosis. We see this in types of white blood cells called macrophages. They may also be called phagocytes. Here's an example of a bacterium, and you get the pseudopodium, so in other words, this extension of the cell membrane to engulf, wrap around, and pull in this bacteria cell. Now, this happens, as I said, very commonly with white blood cells, etc. And what happens is now the pseudopodia fuse at the tips to form the phagosome. Yeah, we know that. Then we send out the lysosome. The lysosome fuses with the phagosome, and it dumps its enzymes inside to destroy the bacteria cell, but it will also break down a lot of the material. The nutrients, such as amino acids, fatty acids, carbohydrates, etc., will be released through the membrane. What is not digested, this vesicle will eventually fuse again with the cell membrane, and you have exocytosis, the release of this or ejection of this material. Now, exocytosis vesicle can be formed inside of the cell that contains materials to be released outside of the cell. For example, not only are you talking about materials such as the release of waste, but hormones, mucins. Later on, we're going to talk about the goblet cells. Goblet cells basically are single cells that produce mucins. When you mix it with water, yep, that makes mucus, and you see them very commonly in the respiratory area as well as the digestive tract, okay? Hormonal cells, cells that make hormones, will manufacture the hormone inside, put it into a vesicle, and the vesicle will eventually fuse with the cell membrane, and you will release the hormone by exocytosis. Now, we've been talking about cell structure, membrane transport across the cell membrane. Now we're going to talk about, how come I'm made of like billions and billions of cells as opposed to one cell? Or better yet, how is it that I don't have the cell, like the old B movies, B grade movies, being they were kind of like, they were cheap budget movies in the 1950s, the cell that took over New York City, or the cell that was 50 stories high. You can't have a cell that big. You can't have things that big. When you talk about an elephant, what's the difference between me and an elephant? It just has more cells. It's bigger. It doesn't have bigger cells size. It has just more cells that make up that elephant. Okay? So we have to get used to certain terminology. When we talk about the life cycle of a cell, we're going to talk about how it makes copies of itself. The life cycle of a cell is called the cell cycle. Now, cells do die. Some of them are programmed, but we're going to talk all about that. When we talk about cell division, particularly we're talking about mitosis. This is a form of cellular reproduction. Apoptosis is programmed cell death. Due to physical wear and tear, toxic chemicals, temperature, environmental stresses that damage the cells, they can trigger a mechanism inside, and the cell will, in essence, cause self-suicide. Okay? And it's gone. But on the other side of the coin, how do I make replacement cells? This is where we get into the basics of mitosis. This is a cell division process that makes daughter cells of all body cells to have the exact number of chromosomes as the mother cell. So in other words, you start with one cell. Let's take a human cell. Has 23 pairs of chromosomes. In mitosis, before it happens, though, we've got to make a copy of all of those 23 pairs. Then we start going through the process of separating out the chromosomes, putting them into different areas of the cell, and begin to pinch off and selectively branch apart, separate apart. What you started with one cell, you now have two cells. Each of the cells are called daughter cells, okay? And each of the daughter cells has the same identical number of chromosomes as the mother cell. Now, there is another type of cell reproduction, but it's only used to make gametes. Mitosis makes cells for all other parts of the body. Gametes are the reproductive cells. Males make sperm, females make ova, or egg cells. We'll get more into this as we get into chapter 26 later on. But let's talk about the cell cycle a little bit more, which really consists of interphase and mitosis. Here we talk about mitosis is cell separation. Start with an original cell, cell division, daughter cells. Yay. Here is the cell cycle, which consists of interphase, and there's several subsections of interphase, and then mitosis, which will start off with the cell, which goes through a separation process. Here we have what is called the metaphase part, where we're lining up all the chromosomes to separate them. Once we separate them, then we begin moving all the chromosomes to the opposite sides and begin to pinch off the cell in the middle to make two separate cells. At that pinching off point, that's called cytokinesis. And this is how we make a replication of the original mother cell. Now, there are critical interphase phases, if you want to say, and this is part of the life cycle of a cell. Let me start right here at what would be called G1. It also, if we stop at G1, we go into what is called G0. What's the difference? G1, usually the daughter cells have just finished mitosis, they've finished separating, but now what's going to happen is they're going to have normal cellular function, normal cell biochemistry. They may undergo some further growth and development. They will make some more organelles. They may have to make some more proteins. There's a certain waiting period. Some cells have very, very long G1 periods. Others do not. But what's a G0? Well, G0, as I mentioned in the lab, it's almost as if the cell has got the engine running, but you're still in idle or you're in park and the engine's warming up. Have you ever had that where you drive or you're going to go out and it's snowy and cold? You got to, quote, warm the engine up. So you get the heater going and everything else. So the car's running, but you're not moving anywhere. G0 is a state where the cell is doing all its basic biochemical functions, etc., but it is in no way preparing or going down the road to prepare to undergo mitosis. Neurons are notorious for doing this. Some neurons will go quite a long period of your lifespan in G0. Some other cells do the same thing, but I digress. Let's say you get certain signals and you have to move from G0 now to G1. Fine. As you go through G1, you have certain basically checkpoints. And if you move through the checkpoint, you will then start to undergo S. Now, what is S? S is when you really have DNA replication. You are going to increase your synthesis of histones. Because remember what I said, if we start with a human cell, we've got 23 pairs of DNA. 23 pairs of chromosomes, excuse me. The chromosomes are made up of DNA and histones. Okay, fine. So if you think about it, it's going to take some time to copy all the DNA of those 23 pairs. When you're complete and done, you're going to have now the DNA for not 23 pairs, but 46 pairs. They're all identical. And by the way, doing this process of the DNA replication, remember you got to have what they call proofreading. In other words, if you make a mistake, put an adenine where guanine should have been. Some of the proteins will correct that error, kick it out, and put in the right one. When you get done with S, which takes a large amount of time, if you notice, you go into the G2 process. G2 is the final process before you start mitosis. G2, you're going to do some selective protein synthesis. One of the selective proteins that's well known to be made in a great amount of quantity is tubulin, because tubulin is going to be necessary to make the spindle fibers that will separate the chromosomes as part of the spindle, which we'll get into in a few minutes. Now, to help you to remember the different stages of mitosis, remember PMAT, prophase, metaphase, anaphase, telophase. Now, by the way, you're going to notice down in here, and I mentioned it before, cytokinesis. Older biology books would say cytokinesis is the fifth stage, or it's part of telophase. Well, that depends on the cells you're looking at. Your cheek cells differ from making blood cells, differ from making muscle cells, differ from making liver cells. When they started looking at mitosis across the different forms of animal life and across the different structures in the body, they found that some of them undergoing mitosis had a different start time for cytokinesis. Sometimes it started early in anaphase. Remember that cytokinesis is the actual separating of the daughter cells. That's the key point. You've already doubled the amount of DNA. You've already put it to opposite sides of the cell. Now it's a matter of separating the cell into two separate cells. But cytokinesis, depending on the cell type, can occur in certain parts of anaphase or telophase, as long as you get that. Now, as I said, DNA replication occurs in S phase. Why so long? Error correction, that's 100%, because if you get a mutation, that could be fatal. How does this process really occur? Enzymes play a critical function, because what you've got to do now is you've got to take DNA, free it from its attachments of the ribosomes, unwind it, and then you got to separate the two strands. When you begin to unzip the double strands of the DNA, you have a particular enzyme called DNA helicase doing that. Once you get going with that, now you've got a simple strand, and you have a unique enzyme called DNA polymerase. It makes the polymer of DNA. What it does is it uses one strand as the template, knowing that you're going to have the bonding to the correct type of nucleotide. If there's a G on one side, you're going to have to put a C right next to it. If you have on the original strand an A, you know that you're going to have to add a T on the new strand. So what happens is you're going to end up forming what they call the leading strand and the lagging strand, which I'll get a little bit more into in a minute. But you want to keep in mind a couple of other important points. Covalent bonds between the nucleotides on the same strand occur. That's because you've got that sugar phosphate, sugar phosphate, sugar phosphate backbone. Hydrogen bonds, which are very weak now, are between the strands of the nucleotides. In other words, basically what holds the ladder or the double helix together, the two strands, is hydrogen bonds. And one other little problem. The DNA polymerase goes in a certain direction, what's called 5', 3'. So that you're always going to have one strand that's constantly being exposed as you separate, you spread apart the double strands. But you got this other one that only shows up a little bit. So what happens is you have so much of the DNA making, the DNA polymerase making some of the new strand, pops off. Make some more of the new strand, pops off. Wherever you have these splicing that's necessary of these lagging strand pieces, you have a unique enzyme called DNA ligase. So I've been talking about this for a few minutes. What's the end result? Remember I said that one strand would be the template, and then you'd have the DNA polymerase making the replacement strand. So you have one strand is original, one strand is a new strand. Here's what I mean. First off, DNA helicase is here, and it's separating the two strands. And this is pretty easy because this is the leading strand. So as we continue to separate them, you've got this nice enzyme, DNA polymerase, just following along, almost like Pac-Man, walk, 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 walk. If this is A, then if this is A, then this is going to be T. If this is G, this is going to be C. On and on and on. But you've got this other strand, and DNA polymerase can only go in 5' 3', so that means you can only go here and go in that direction. Problem, you can only go for a distance of about 1,000 or so base pairs. You would have this base and these two bases together. That's called a base pair. And then it pops off. No problem. You're going to get these smaller pieces of strand on this side. We'll have a DNA nucleotide that's inserted in here, patches it together with a unique protein called DNA ligase. In other words, you always have got to have your backbone here that's held together by covalent bonds and that is continuous, because the stability is partly due to the covalent bonds between sugar phosphate along the backbone. Okay? This would be the lagging strand. Now, I've just given you kind of that basics of DNA replication, but what we really need to do is start talking about the steps of mitosis. Now, mitosis is really the division and duplication of the cell's nucleus. Cytokinesis is really just the cytoplasm divides into two separate new cells. Now, you got to keep in mind that prior to mitosis, before we start mitosis, the PMAT, the DNA strands are been loosely coiled and the chromosomes cannot be seen yet. But once we get into prophase, you're going to get what is called condensation. In other words, the DNA strands that were loosely coiled up are going to be tightly coiled up, wrapped up with the histones, recoiled, and that's where you're going to get the formation of the chromosomes. But each copy of these chromosomes are called a chromatid. You have what is called a kinetochore, which is the raised site on the centromere where the spindle fibers will attach. Wait a minute, what's the centromere? The centromere is sort of this nice little attachment point that really holds together the chromosome and its copy. Okay? Remember, if I had 2 copies of chromosome 1 and I go through S phase, I'm now going to have 4 copies. I'm going to always have an original, and I'm going to have a copy that's made. So the original and the copy get held together by a centromere. But then there's an extension off the centromere called the kinetochore where the spindle fibers attach. We're going to talk about the spindle fibers in a second. Spindle fibers are really microtubules formed between the pairs of centrioles. Centrioles replicated, there's 2 of them. They move to the opposite sides of the cell. This is the beginning of prophase. They have to move to the opposite sides, and we call that the poles of the cell. Is that sort of like what we have poles on the Earth and we have an equator? Yeah. Because when we get to metaphase, we're going to line up all of these pairs of chromosomes in sort of a nice linear shape. And that's why we call it, they form an equator-like plane inside of the cell. Then we get to anaphase. And that's where we separate the chromatids. So chromosome 1 goes to one side, and chromosome 1's copy goes to the opposite side. How are they pulled apart? By the spindle fibers. And they're going to pull them in the direction toward the, basically the centrioles. Here's what I mean. We're starting with mitosis. We have these astral arrays and spindle fibers, but what's at the center here are the centrioles. They're going to eventually spread outward. Now, how can I get at the chromosomes if they're encased into a nuclear envelope? Real simple. I dissolve this nuclear envelope. When you dissolve the nuclear envelope, you start seeing sort of a free motion or an easier access, someone has spacing between all the individual chromosomes. Now, if you look at this cell here, you have now the spindle fibers radiating all the way out. Now, by the way, what astral rays are really are those that are attached. They're spread out opposite to where the nucleus would be. So these would be the sort of astral rays here. These are the astral rays here. The key point is that for each and every one of these chromosomes, they're going to have an attachment to a spindle fiber, okay? And they're going to be attached at the kinetochore. Now, the chromosome and its copy are called chromatids, okay? We get them all nicely lined up here. This is the metaphase plate, otherwise known as the equator. Here are my poles. This is metaphase. The next step, and there are videos that I provide for you to review this. You can see this in time-lapse photography. It's really awesome. When you move into anaphase, what happens is the spindle fiber apparatus separate out the chromatids, the original and the copy, and they go in different directions. And you see how they're kind of bent, almost look like forks? Well, think about it this. You're being pulled through the cytoplasm. So the area that's going to be sort of like at the very tip attached to the spindle fiber, that's going to be the leading part, and the lagging parts are the edges that are kind of dragged along as you go through this cytoplasm. So they each go in opposite directions of the opposite poles. Once you get this done, you're finished with anaphase, and now you just go into telophase, which really happens is that in the process of telophase, you're going to take and envelop those chromosomes in a new component of nuclear membrane, okay? And you're going to start breaking down the spindle fibers. You don't need the spindle fibers anymore. But what's going to also happen around this time is you're going to start forming this cleavage furrow. And this is basically a pinching off in the center. And that's part of the cytokinesis. So you separate out the cytoplasm. Basically, you make two cells. They're called daughter cells now. And you can see these here. Okay. So telophase is the cell prepares to enter interphase. The new nucleus envelopes, basically they're going to envelop the chromosomes. The next step you're going to see is the chromosomes uncoil. So you'll now have more chromatin, okay? And in a short time, you're going to be able to start having gene reading, gene transcription, making proteins again, everything. Cytokinesis, the cells are separated. Now, by the way, so I help you to understand this now. Many cells cannot have more than one nucleus. Everybody got that? Make sure you write that down. It is, nature hates too much or too little of DNA in a cell. Then you end up with all sorts of haywire activity, etc. But in a few unique cases, very few, as we get into tissues, you're going to find that some cells, such as a skeletal muscle cell, can have more than one nucleus. So what happens there? They just don't have cytokinesis. They do have mitosis. They separate out and have another nucleus made, but the cell does not divide. But that is the exception to the rule. Make sure you're clear on that, okay? And then, of course, we talked about cleavage furrow. The cytoplasm constricts, pinches inwardly. And this is basically done as almost like a basically like a string purse. Pull the string in, the opening of the purse closes inward. You've got this pinching inward that separates the daughter cells, and the mechanism is partly done by the proteins of actin and myosin, okay? So we've been talking about all the normal cell behavior, and we do have to get familiarized with a couple of terms talking about abnormal cells. Everybody thinks of cancer. Cancer is an illness caused by mutations that eventually lead to the cell no longer controlling its own division rate. Now, what you have to think about is it's not just a matter of the cell turns weird or does certain weird things. It's the fact that the cells do not control the division rate. You can have them just constantly dividing and dividing and dividing. Now, mind you, as they're doing all of this, you're going to have abnormal chromosomes. You're going to have also a greater demand of nutrients. Wherever the cells are growing, they may grow in a solid mass or they may be diffuse throughout a particular medium, such as blood. So we get things like leukemias and lymphomas and things like that. But when we talk about mutations, these are changes in the DNA sequence. We actually have oncogenes, genes that if they're damaged, they trigger the formation of cancer. So we get the word oncology, onco meaning cancer. But what happens is some mutations are silent. They don't cause any changes. Other changes can cause radical differences in making proteins or in controlling the life cycle of the cell. Now, when we talk about the term tumor, this is also referred to as a neoplasm. It's a mass or swelling due to an abnormal cell growth and division. There are different types of tumors. One is called benign. This is where the cancer cell will remain in the originating tissue. It can be removed and the patient cured. When we talk about malignant, though, this is a cancer that generates chemicals to stimulate new blood vessels at the original site, and cells then migrate into the surrounding tissues. A lot of times they go via the blood vessels, but there's sometimes that they will migrate not using blood vessels, but using lymphatic vessels. They'll show up in lymph nodes. They will travel to other parts of the body via the lymphatic system. Okay? Metastasis is the process of a malignant tumors producing secondary tumors remote from the primary site. Think of it this way: if I start with a stomach tumor, the tumor starts growing, and eventually what happens is the cancerous cells start basically moving into the blood system and then go into other areas, such as my lung or my bone or other parts of my digestive system or up into my brain. That's metastasis. The cells have gone and have begun to travel in other places, and therefore treatment is necessary not only to remove the original tumor, but either remove the other tumors or to treat the body to prevent the secondary tumors from growing further. Okay? Usually those treatments include either radiation or may involve very powerful cell chemicals that we call chemotherapy. They may be acting on unique biochemical properties of that tumor. Here's a situation of an abnormal cell. As you can see, this has begun to mutate, and it now has lost its control for mitosis. And so now what we have is a lot of cells dividing quickly here. And as you can see, they're starting to press upon the underlying tissue. They're nearby a blood vessel, and they definitely have erupted beyond the limit of what these original cells here. If you look further, the tumor, which was primary, has now had cells that now have the capability to migrate. A lot of times when they have invasions, the cells now have developed unique properties. Some of them have developed enzymes that will allow them to break down surrounding tissue so that they can get into and spread into the bloodstream. Others may release angiogenesis factors. In other words, chemicals, usually proteins, that basically will turn on the development of new blood vessels to feed the primary tumor. And also, it may be the secondary tumor. What will happen with these cells is they may travel throughout the bloodstream, eventually escape, and get into other tissue areas where they will begin to develop. Okay? As you can see here, we are now having the development of growth of blood vessels into this new tumor that will then be fed. One of the techniques that's been used to block tumor growth has been to try to find chemicals that will block what they call angiogenesis, the formation of new blood vessels. And in doing so, the tumor growth is stunted. The cells grow at such a rate have such a high metabolism that they constantly need being fed and thereby need really close or actually be invaded through them with blood vessels to supply them with their needs of oxygen, nutrients, waste removal, etc. Okay? Here's an example of thyroid cancer. Now, what has happened here is that the thyroid cells have gone haywire. Unfortunately, this poor individual has not been able to be treated early, and so the tumor, this mass of cells, has grown excessively large. One of the other things that happens when we study the thyroid, which will be an AP2, is that it has a very rich blood supply. It's necessary for that. One other thing I think you ought to know now, and you may want to put it as a side note, is that usually when a cell goes haywire, goes cancer, it takes along some of the properties of the original cell that started off. It's very common, for example, endocrine cells. Let's say those that are in the medulla of the adrenal gland or the thyroid or other particular ones, the pancreas. If they have been originally making an endocrine secretion, in other words, a hormone, now, once they have gone cancerous, they will be usually producing a much higher amount of the um, hormone and usually be not responsive to the body's feedback mechanisms to stop the secretion. So it's almost like they're turned on and they're always turned on. They will not turn themselves off. They will not regulate themselves, which is another problem because one of the symptoms sometimes of being able to discover the presence of cancer is the excessive secretion of certain hormones. And then that leads the doctors backtrack to the point of where and what the tumor is. Okay? Now, in this case, to treat this individual, they would probably do surgery. If this individual was in an area that would allow it, they might get also radioactive iodide to basically shut down a lot of the process of any remnant cancer cells that were present. Okay? All right. And we're done. Okay, so this is chapter 3. So I encourage you to peruse the chapter review pages on 137 to 139, complete review questions on 139 and 140, complete the review sheets on pages 108, 118, 128, and 136. Now, this is for your own edification. This is for your own review of your progress. Do you really understand what you're reading? You don't send that to me. What you do is that if you find you can't answer some of those questions, one of the best strategies there is stop, put everything down, and go over and study that content that you don't understand yet. Because this will help you self-diagnose what you don't know and get you better prepared for the subsequent chapters as well as, of course, the exams. Now, the next lecture we're going to be dealing with is chapter 4. This is the tissue level of organization. And I hope you have a great day.